Well, good afternoon and welcome to MedTech Crossroads. Today is Friday, August 21st. Today is the 22nd episode. I'm Gene Paranek. I'm so glad to have you here. We have a packed show today. Today we have Dr. Duane Newton, who's the director for the Clinical Microbiology Lab at the University of Michigan. We have Dr. Niki Kennedy back for the second installment of From the Frontlines. We're going to be talking about the felt needs of doctors and healthcare providers in the innovation and healthcare delivery ecosystem. And last but not least, I've got Eric Icard from The Right Place, the big group over in West Michigan and Grand Rapids, does business development and incubation. And he's going to tell us all about the business network in West Michigan that came together during COVID. And they sure got to put some of their networks through the paces, and we want to hear about that. So without further ado, with that packed schedule, I want to welcome first Dr. Duane Newton, the Director of Clinical Microbiology Lab at the University of Michigan. Dr. Newton, you can unmute your audio and unmute your video. And I'd like to welcome you to MedTech Crossroads.
Thanks so much, Gene. Thanks for inviting me. I'm going to get my slides started up here if I can get everything. Whoops. That's the wrong button.
And of course, you guys are used to doing testing there at the microbiology lab.
Yeah, I'm not used to working on the Zoom computer and giving presentations. There we go.
Well, you guys also talk about getting things exercised. You guys got put through your paces recently with the uptick in COVID testing, which I think you're going to tell us about here.
Yep.
Looks like those slides are coming up. Looks great. Take it away.
All right, super. Thanks. All right. So sorry about that. Really, the focus of what I wanted to talk about today is really the SARS outbreak from a testing perspective. I think there's some basic information that is useful. I don't think a lot of people have shared or have been able to provide a perspective on. So I wanted to do a little bit of that with regards to the virus, where it came from, and how we perform testing in clinical laboratories. And first, I want to just give you a little background about me and understand, what I do and how that fits into patient care. I have a bachelor's and PhD in biology. I went to school in Dayton, University of Dayton, and was really on a basic science track for a while, and realized that I was more interested in applied science and more patient care focused, and completed a clinical microbiology fellowship at the University of Rochester. And that fellowship was designed to train me to be a director of a hospital lab. And my first job coming out of there, I actually worked for the state health department in Lansing for a couple of years before I was recruited to come over to Ann Arbor. And I've been here in Ann Arbor since 2002. And I'm on my last couple of weeks in the Department of Pathology here before I embark onto a new path, getting involved in consulting and working with diagnostic manufacturers. And we can talk about that a little more at the end. But I think one of the things, as I reflect back on my career here in Michigan, been involved in a lot of different outbreaks over the years. Started with West Nile when I was at the health department, and there was actually a SARS outbreak in the world before this one. And MERS and pandemic flu and Zika, Ebola, and now COVID. So I've been through this quite a bit, but this was by far the most challenging one to date. I don't know how much people really understand what goes on in laboratories where the diagnostic testing occurs, but this I felt like is a really important thing and to share with you. And this outbreak has really given us an opportunity to highlight the hard work that goes on in that black box that gets called the lab. Really, our lab is one of many labs in the Department of Pathology that performs testing in support of patient care. And the clinical micro lab is performing infectious disease testing. And we're trying to find bacteria, viruses, parasites, fungi in clinical specimens that are obtained from patients that the physician feels like has an infection. We have about 60 technologists in our lab that perform testing. Some of it is very hands-on and manual, and some of it is using the very sophisticated equipment that you see on the screen. We operate 24/7, 365. The patients are always sick, never take a vacation, and we don't either. We have a pretty challenging task that we're held to right now, especially with COVID and the volume of testing that's coming in. They actually don't let me touch any patient samples. I have to stand back and let them do all the work. That's what they're trained to do. My job is really, I like to describe it as captain of the ship or conductor of the orchestra. I have to understand at a technical and a clinical level what's required. I collaborate and consult with the clinicians who might have questions, but I also help at a technical level answering questions for the technologists at the bench. I just make sure that we're going on the right direction, and that we're all on the same page. And in the context of COVID, one of the real important things was to figure out how we were going to accomplish that. And I had a whole team of people that were working on that, and I was a part of that team. In the bigger context of biology, what are these organisms and how do they fit in? I thought it would be good just to put things into a little bit of a perspective from a practical standpoint. On the left, you can see kind of what the size of a red blood cell that's in our circulation. It's about 10 times the size of a normal bacteria that might cause an infection, and it's hundreds of times, upwards of hundreds of times bigger than some of the viruses that cause infection. So just to show you the size difference and how that presents a challenge for us to detect these organisms in a patient specimen. And so all the methodologies that we use are designed to really enhance our ability to see that very small, not necessarily see, but find that very small organism. And so the virus that SARS-CoV-2 that causes COVID is about the size of this adenovirus, and looks kind of similar to that if you would look at it with an electron microscope. So just from a terminology standpoint, you say SARS and SARS-CoV-2 and COVID-19, they're kind of all referring to the same things, but from a very specific standpoint, the virus that causes the infection is called SARS-CoV-2, and that's to distinguish it from the original SARS coronavirus that was identified in the early 2000s. The disease that it causes is referred to as COVID-19, or coronavirus infectious disease at 19. It actually started in China in 2019. Where did it come from? We're not exactly sure, but it's thought to have been of animal origin, maybe bats. And some of the first epidemiologic investigations found a lot of infections that were associated with live animal markets in China, but it's not clear that it was actually acquired from the animals in that market, that it may have been just the congestion of people, and that infected individuals were there and spread it to other individuals. And those live animal markets, both because of unusual organisms that might be passed from animal to human, as well as the very close quarters and interaction between people and those animals, have caused a variety of different emerging infections, and there's very easy transmission. And this virus was very effectively transmitted from person to person. And that's really distinguishes it from some of these other outbreaks that have happened earlier. The original SARS coronavirus and MERS coronaviruses were not easily spread from person to person, and so those outbreaks kind of fizzled out over time. The SARS-CoV-2 is actually one of a member of a family of coronaviruses that we see all the time. There are many, many types of them, and most of them just cause mild respiratory symptoms like a common cold that we always get. But the original SARS, SARS-CoV-2, and MERS actually can cause very severe disease, as you've seen in the news, and hopefully not experienced directly. But the reason why these viruses cause severe disease is because we've never seen them before. We've seen these common cold viruses a lot, and so we've built up some immunity to them, but we've never seen these other viruses before. And because of that, we have no protection, and they're able, especially in patients whose immune systems don't function very well, they can cause very, very substantial disease and unfortunately fatal. One of the biggest things that I've been spending my time on over the last several months is trying to optimize a testing approach to detect this virus in a patient specimen. And to understand how we do that, you have to get into a little bit of genetics and go back to some of your high school biology. But as a refresher, I like to think of how our genetic information is organized. And think of it like a book. And if you think of your book as the nucleus in our cells contain a nucleus that has all the information that we need, that all of our genetic information is located there. And those books have many chapters, and those chapters tell different parts of the story. And I like to think of our chromosomes, which we have 23 pair in our cells. We have 23 pair of chromosomes. Oops, sorry. Then on those chromosomes, in the chapter, there are sentences with lots of words, and those are the genes that tell our cells, what we need them to do. And the letters of those sentences are actual nucleotides, these individual bases that make up the message that forms all that information that's in the book. Microorganisms are not as complex as our cells. This is a cartoon of our cells. Microorganisms have parts of this. They'll have this nucleic acid strand. They're not going to have all of the, they're not going to have all of the information, the amount of information, and have it packaged in the same way. They're going to have shorter sections of this. So while we have millions and millions of base pairs in our genome, the SARS coronavirus has about 30,000 bases that make up its genome. So what we're trying to do in a clinical specimen, we use techniques that actually identify SARS coronavirus sequence, unique SARS coronavirus sequence in a clinical specimen. And we have to find it. It's like a needle in the haystack, finding that in the middle of all that patient material. Another type of test that's available are antibody tests. And those are trying to detect the products of our immune response that are found, that are produced by our immune system. So there are special cells that are part of our immune system that recognize the fact that we've got an infection and produce antibodies, and that's what these little dots are, that will bind to the virus, and then your immune system recognizes that that virus shouldn't be there and will gobble it up. And so another way we can detect whether or not a patient's been infected is to look to see if they've made antibodies against a virus. And a lot of tests that are out there commercially available are looking for antibodies. This generally happens several days to weeks after infection, though. One of the challenges that we had was how quickly we could bring tests up to market. And the biggest issue is that this was a new thing. This was a new disease, and there weren't any tests when it started out. So it takes time. Usually we have months to years to bring a new test to market, but in this we had days to weeks, and that presented a challenge. The regulatory landscape with regards to the FDA was also challenging because the process to get approval was still, even though it was expedited, it was limited to commercial manufacturers and was not made available to labs like ours that could have the ability to make our own tests. And that changed over time and allowed things to happen a little more quickly. But it still took well into March before we had tests that were available, even though there was evidence of this being a big issue late in 2019 and early 2020. The CDC has gotten maligned quite a bit, and I think unfairly, because the CDC's role is to protect public health. It's not to do testing for everyone in the United States. And So, and from a public health standpoint, they'll start the process, share information with public health labs, and then it's up to the private sector to pick up the ball from there. And for some of the reasons that I talked about before, this was new and the regulatory landscape was challenging. It took a while for that whole process to get rolling. But the CDC was not and is never intended to be the answer to these problems. They start the process and help us get going, but we have to pick it up from there, especially in an outbreak as big as this. And then, one of the things that we continue to deal with is that as the number of cases and the number of people that needed to be tested increased, the availability of supplies went down very, very quickly. And so we still are struggling with that on a daily basis. And it's not only COVID testing supplies, but it's others. These manufacturers also make other stuff that we use in our lab, and we're having supply chain issues for that as well. Now, I just want to end with a couple of important points to understand that what this cartoon is showing you is the normal course of disease. And the red line here is a person who's been infected and when they start to show symptoms. And so when they become infected, they're not symptomatic, but over the course of 5 to 10 days, their symptoms will start to peak. And that the correlation of the peak symptoms is also the correlation of the peak amount of virus that's being shed in their secretions that we can detect. So the assays that we use that are specifically detecting the virus are going to perform the best when the patient is symptomatic. The assays that are designed to detect the immune response are indicated by the green and yellow lines, and those are antibody tests. And as you can see, they don't start coming up until well after infection, 5, 10, 2 weeks. And overall sensitivity of diagnosis is shown here with the dotted line. And so over time, you can improve your diagnostic capabilities of being able to detect infection in a patient because they've had a chance to have a lot of virus produced or a lot of antibody produced. So what the red shows is that there are actually times when tests can be negative. And for the tests that are detecting the virus specifically, that's in their pre-symptomatic or asymptomatic phases before they're starting to produce a lot of virus. So upwards of a week of infection, we might miss that test because there's just not enough virus to detect it. And then as the patient recovers, that amount of virus is going to go away, and we'll start to not be able to detect it again. What we know right now is that the antibody tests come up within a couple of weeks and stay around for a while, but we don't know how long they really stay around. And so what we've seen is that there is some period of false negatives after a patient has been infected and recovered where even the antibody is not going to be detected. And we also don't know a whole lot of information about how protective those antibodies are from a repeat infection. But the premise is that they will protect you, and that's the basis for all the vaccine development, is to stimulate the antibody response so that you be protected from an infection. So with that, I'll stop, and we can move on to whatever Gene says is the next way to go. If you want to do questions first, or the next speakers, and save questions for the end.
No, we'll do questions here in just a second, and actually I had a couple, but to our audience, if you have a question for Dr. Newton, please do raise your hand in Zoom. We'll get to those in just a second. Feel free to raise your little Zoom hand there if you'd like to ask Dr. Newton a question. This is wonderful, and every time we see one of the graphs like you showed on the last slide, it just shows us more how you referred to people thinking of labs as black boxes and how they really aren't. And it's just, if there's one thing that we all need to learn out of this, It's the nuance that goes into this. These are not simple binary yes, no answers. And we've learned that every time we've had someone who's in biology come on and explain some of the details. And I love your graph. There's very succinct, showing the serology and the RT-PCR, kind of how they overlap on the same graph. One question that came to my mind as you were talking, of course, now is, and I don't even know if when you and I first chatted, if this had even come out yet. NIH is talking about some evidence that T cells are showing some level of, I probably won't say this right, but they're showing some level of having seen some sequences before. The theory then being that it's on the basis of previous coronavirus, but not not SARS-CoV-2 infections. Can you speak to that at all in terms of this interplay between the antibodies and the T cells, and do we have different mechanisms going on here under the hood?
Yeah, you're making me call back to those times when I was back in the University of Dayton and doing my basic science research and that studying the immune response to infection. So you have two arms of the immune system: the cell-mediated part, which is organized, managed, moderated by T cells, and the antibody arm of the immune system and the immune response, which is B cell mediated. And so there are certain pathogens, and a lot of viral infections are actually managed because of T cell mediated response. And you have cytotoxic T cells that recognize virus infected cells. So it's entirely possible that and I'm hypothesizing because I don't know the science behind it, but it's entirely possible that the immune system does have a major part, being T cells being responsible for dealing with this with this pathogen, and that may be a reason why the antibody aspects of things are not very. It's not very clear. It's not clear that the antibodies aren't staying around very long, and it's not clear that they're very protective. And so, I'm not sure exactly where. I looked this morning. There's about 200 different vaccine trials going on, and I don't know how many of them are designed to stimulate antibodies and how many of them are designed to stimulate a cell-mediated response. But it'd be interesting to kind of look at that and compare and see, what direction they go and which ones are most effective.
Yeah, I think the biggest thing for a lot of folks to just recognize is that there are so many different levers to be pulled underneath the hood. There's so many different ways that these things work underneath, and people tend to think in very, very binary terms. And so it's good to just unpack a little bit of that. We've got 3 questions here. Let me go first to our friend Ken Spencer. I'm going to make him live, and then after that, Brandon and Olaf, I'll bring you guys live if you want to ask your questions. But Ken Spencer from MCirc over at U of M.
Yes, Dr. Newton. So I work over at MCirc for Kevin Ward over there in emergency medicine. And I was wondering if you were familiar with Dr. Scott Van Epps' work on a rapid nanorod PCR test that he has a project going on there.
Yeah, Scott and I are collaborators and friends. So we've been working together on a variety of different projects, and he asked me for samples, and I graciously give them to him, and then he does his magic with it. So I haven't had discussions with him about that particular technology, but I am familiar with it. And I think, one of the things that he's very interested in, and a lot of investigators and diagnostic companies are interested in, is trying to figure out from a technology standpoint how we can make these tests simple, fast, cheap, and reliably insensitive. And so, there's like all these magical components to it. And a lot of what I know what Scott is working on is to try to accomplish a accomplish all of those things in detecting infections. And so the stuff that he and I have worked on have been related to bacterial infections.
Great, thank you.
You bet.
Great, thanks, Ken. Next up, got Brandon McNaughton.
Duane, it's good to see you, and it's nice to see your presentation.
Hi, Brandon. How you doing, buddy?
Good. Just been busy at the normal work of working on new products and technologies. But with that in mind, one project we have is on trying to be able to pool saliva. So we have a concentration method for that, and we have an active project, doing that with saliva and see if we can overcome dilution. So I was curious to get your thoughts. I mean, certainly increasing throughput seems to be a big issue right now. But with the opportunities in developing new tools for diagnostics, where do you see the biggest challenges or opportunities? I mean, you mentioned them there, the simple test and others, but are there other areas or problems and pain points you see right now for testing?
Yeah, Brandon, I think that's a really good question, and I think very timely in the context of the recent news coming out of Yale with their saliva test, and that's getting a lot of publicity and maybe a little bit of information that's not entirely accurate. And so saliva as a specimen, and the studies have several studies have shown that it's less sensitive diagnostically than a nasopharyngeal. A nasopharyngeal swab still remains kind of the gold standard, and it's been shown multiple times. But I think depending on the patient population you're talking about, and if you go back to that graph that I was showing where the person is in the course of their disease, a less sensitive method or a less sensitive specimen type can still be very, very useful. And saliva as a specimen, easy to collect. The types of PPE that a healthcare provider that's in the presence of the patient would need to use is much less, because the aerosolization risk is low. It's not zero, but it's not the same as collecting an NP swab. And in fact, people, you can do self-collected swabs or self-collected saliva. So saliva adds to, or enhances the ability to easily collect a specimen. But I think, and so that overcomes a major challenge operationally, which is the availability of the collection supplies and PPE. So that's a huge bottleneck right now that we, on a daily basis, are dealing with. But the second bottleneck is really throughput. There's so many specimens that need to be tested, and there's so many people, so many places, institutions, organizations that want to have testing right now that it's very, very difficult to provide testing to support patient care, as well as testing to support opening of schools and opening of businesses and other areas of healthcare like the dental community. And so there's just that burden or that demand it is overwhelming the capacity. And so I don't know how long that's going to continue, but that's certainly something that we're challenged with. And the idea of pooling specimens to increase your throughput is a very, very attractive one, but we can't do that and lose the sensitivity that we expect for the patients that are being tested. Because for us, what we would be doing is pooling all of our asymptomatic screening specimens, which are very high volume. And these are patients that are coming in for pre-procedural, and clinicians are not wanting to miss that 1 to 5 in 100 that you would miss at a minimum by using a less sensitive specimen and by pooling it. So if your approach to enhance detection of the target increases that, or maintains the analytical performance and increases the throughput, then that's a big winner, Brandon.
That's what we're working on. I'll follow up with you about the details.
Okay.
Great. Thanks, Brandon. Our good friend Olaf has actually three questions. I want to give him a chance to actually ask them all. So we'll go kind of quick on the question and the answer for these, but he's always got such great questions. Olaf, you're live.
Okay, yeah, I thank you. Dr. Newton, thank you for taking the time and these really good explanations and presentation.
My pleasure.
Is about the detectability. So I understand that, antibodies are developed later. It takes time for the immune system to respond. But I understood from what you said is that even when somebody is infected, let's say, at time 0 hours, that it can't be detected for the first number of hours, but it takes, maybe even several days to be detected. Can you explain that? And is there something about, you mentioned the sensitivity of the tests that would help there?
So that statement is true for both antibody tests and the molecular test to detect the virus. So I'm going to see if I can pop my screen back up here and just show that graph again. So can you see that, Gene?
Yes. Yep,
Okay.
You're just fine.
So virus is circulating in the community and causes, gets transmitted to a person. And once it starts to invade our body and starts replicating, that's time 0. And it does it on a very small scale initially, and then it spreads from cell to cell in our body. And that's what causes us to have our respiratory symptoms. And so that is the blue line here. So it's just that delay is just because it takes a while for the virus to infect enough cells in our body for it to have those detrimental effects that we start having. The symptoms that we feel and express are because of the amount of virus that's replicating in our respiratory tract.
Interesting.
And so sort of concomitant with that is our immune system trying to fight it off. And that takes a while to happen as well, especially for something that we've never seen before. If we've seen it before, our immune system has memory and it kicks in a lot more quickly. It should kick in a lot more quickly, or it can. It has the ability to for many things. But with this, where we've never seen it before, we're naive, and so it takes time. And it's just sort of the natural biology of our immune system is that an antibody response doesn't happen for a week to 2 weeks. And not only does it not happen, but it doesn't happen to a level consistently that our tests can actually detect it. And so, there are thresholds of the amount of virus or the amount of antibody that needs to be present before our tests can actually see it. And so, one of the things that we want to improve is the analytical performance. How much virus or how much antibody needs to be there for us to be able to detect it. And so that closes that window and makes that, this will be gray a little bit further to the left if we can do that.
I see. Right. Okay. Okay, great. Thank you. So my second question is kind of similar to that. So what you've mentioned is essentially the fundamental detectability of the virus itself with a molecular biology test or of the antibody.
Mm-hmm.
How about, we often also on the show talk about false positives and false negatives. Is that also something that changes over time? I guess, is the detectability kind of equivalent to false negatives? So
yeah. So that's really a great question, and it's a difficult and nuanced answer, and one that we have to address frequently in a clinical setting with our physicians to help them understand the performance characteristics as well. And so there's definitely false negatives, and we talked about that. So the beginning, before you can detect anything, and then as the patient is resolving, the virus is going away as well. And so they may have been infected, but now we can't see it anymore. Antibodies should help with that, but it's not perfect as well, because we're finding that these antibodies don't persist forever and they start to go away.
Yeah.
So that's the false negative thing. False positives is a much more challenging question to answer. So there's analytical false positives, and that's the target. We detected target, but it wasn't really there to begin with, and that's a lab problem. Okay, so we contaminated it, or maybe the specimen got contaminated, or if you're theoretically, if you're not careful with how you're collecting the specimen, you can get patient material on the outside of the container and not know it. And in the process of handling a positive patient, that material can be transferred to other patients that didn't have an infection. So that's called laboratory contamination. That could happen, but we take lots of precautions and have quality control measures in place to show that that's not happening routinely. So analytically, we shouldn't have, it should be very, very infrequent that a positive result is from that kind of a scenario. What's more likely is that we're picking up positives in patients where their symptoms are gone. And our test, our molecular test to detect the genes are very, very sensitive. And so this graph of symptoms may actually kind of like drop off more quickly. And this blue line of virus being detected can kind of tail off for a while. But we don't know if that is really infectious virus or just nucleic acid that's persisting after recovery. There's a lot of evidence in other infections that our molecular tests will be persistently positive, even though the patient's recovered and responded to therapy. So that's not an unusual thing. So that I categorize as a clinical false positive. We detected it, that target was really there, but it doesn't correlate with what's going on in the patient.
That's great.
Super.
Just looking as a question of time here, I just want to be sensitive to our other guests on the show today. These are absolutely phenomenal. There is one more question, and Dr. Newton, you ought to be able to see Olaf's third question in the Q&A chat. I'm wondering if you could take a look at that and place a response. But I also do want to give you one more moment to tell us, because you're making a big career change here in the next little while that you alluded to. I wanted to give you just about a minute to tell us about, I think it's called NaviDX, that you're going to go join.
Sure, yeah, thanks for the opportunity to share that information. So I've been in clinical lab for 20 years plus my training, and you have had the opportunity to work with a lot of companies in the context of that and clinical trials as they've had new products being developed. But I've also consulted on the side with companies that are in various stages of development. And actually, Brandon and I first met as he had a startup coming out of the U of M from the technology that he developed, and was helping him work on bringing that product to market. And I've really enjoyed doing that over the years, and got to a point in my relationship with NaviDX that the opportunity presented itself that I could do that full time. And so I'd be working, they're a small consulting firm that has expertise in a wide range of areas of diagnostic manufacturing and commercial development. And I'll be the clinical microbiologist, the patient, the patient care perspective, the clinical lab perspective, that to add to the business and technical development perspective that consulting company has. And so hopefully, be able to continue to improve patient care, but just from a different pathway, and intervene in the development process early enough that can help fill some of these gaps that we see in the lab.
That's wonderful. We wish you the best in your new career path. I don't know if you're willing to share some contact information for the guests on the show here today, but if you are, you could chat that to the panelists and attendees, because I know we didn't quite get to all the questions, and whether it's on the University of Michigan lab side or the NaviDX consulting side that's coming up, I'm sure folks would love to be able to get in touch with you.
Yep, I'll get my Gmail address in the chat to everyone.
Well, we greatly appreciate it. Dr. Newton, this has been such a pleasure. Thank you for doing that. I have one more question for you. How many tests, if you're able to share, are you guys doing, or have you been doing, or did you do at peak at UVM there?
I peaked yesterday, and we're right about, since we went live with our internal COVID testing on March 17th, we've done just about 90,000, a little over around 90,000 tests. And so that's 2.5 times the highest volume molecular test that we had in place annually, for our annual volume that we've done in those few months. So it's been a huge impact on us.
Well, that's wonderful. Thank you for doing that, and thank you for being on the show with us today and for sharing this with us. As we surmised, lots of questions, good questions around this, and we appreciate all the work that's going into it. So thank you for being here.
Time.
Thanks again. We appreciate that. So again, like we said on so many shows, it's so good to see so many folks around the state doing so many amazing things in response to COVID. Next up, I want to welcome Dr. Niki Kennedy of ArborHive for the second installment of our From the Frontlines feature. And we are so glad to have Niki Kennedy with us. Are you there, Niki?
Hey, Gene, can you hear me?
There she is, Dr. Niki Kennedy with ArborHive for the From the Frontlines feature, our second installment. Niki, it was about 2 months ago or so that you and I were talking, and we said, boy, we need to hear the reverse pitch from doctors about what they run into every day in their work. And you said, well, I can do this. And so ArborHive and MedTech Crossroads said, well, let's do this together. And this has been phenomenal because last time we talked about electronic medical records and insurance payer systems, as I recall.
Yes.
And we had 2 more topics. One was compliance and one was communications. And as we said about a month ago, all of my startup friends are going to go, well, you didn't talk about my product or the great need for my product that every doctor feels the felt need for. And I think that's part of the reason why we're doing this is because we're trying to sit back. Those of us who are involved in product development, we're trying to just sit back and listen because you're in a wonderful position. You're an engineer, you're an MBA, and somehow along the line you became a vascular surgeon. And how that happened, who knows, right?
I'm still trying to figure that one out.
And you've had a great opportunity with ArborHive, which especially physician innovators can go join to become part of a network and to learn more about innovation and development from a physician's perspective, which I think is just so cool that you're doing that. You've had an open door to talk with many of your friends and colleagues and say, what are your felt needs? And what are we going to talk about today?
So I know you and I have a lot that we could talk about, and usually when we get together we do, but I know you've got some limited time. So I was thinking, let's talk about some of the communication issues that we find today. And again, if we need to do another installment at some point, we can set that up, but let's see how far we get. I will say my usual disclaimer, I do work for a health system, but I do not represent any particular health system or speak on behalf of any health system. This is all purely my opinion and what I hear from my colleagues. So, and last time, as you said, we talked about some of the problems that physicians see, and I thought you did a great intro right now there, Gene, because we're looking at a lot of problems that, again, may not pertain exactly to somebody's device or new software as a service platform, but hopefully they all kind of fall under an umbrella or hit into a bucket, as you and I had talked about. And one of those buckets we'd referred to when we were first coming up with this was the idea of communication gaps. I think every industry and every place can see a communication gap somewhere along the way. In the physician's lifestyle, we find that in a few different places. One is going to be with respect to how we communicate with our patients. One is going to be with respect to how we communicate with other caregivers, other physicians, other colleagues, and then also how we deal with offsite services, ancillary groups like physical therapy, supplies and durable medical equipment, those sorts of things.
And so I think when people think of communication, and I think I might have just lost my video. I'm not sure if you can see me, Niki, or anyone else can see me.
You look a little frozen. I just wanted to pause to make sure you were still there.
Thank you very much. Well, my video will come back, but as long as everyone can hear me, we'll have folks work on that. When we think of communication, oftentimes, I don't know if people really understand what types of communication people on the front lines are up against. So walk us through this. Where do communications breakdowns come, or where are the challenges? Because well, I guess many people who are focused on relationships and stuff would sort of say, yes, communication is very important. Many of the rest of us just go, well, I don't know, I talk and stuff happens. But in a healthcare system, lots of balls can get dropped all over the place. So where are some of those places?
So the one thing I've learned from doing the MBA and working with other groups is that there are a lot of things that we're trying to accomplish. If we're working from a startup standpoint, we want to make sure that we're going to be getting our money back for our investors. If we are in advertising and marketing, we want to make sure that we're getting sales. But in healthcare, the end result, despite all these other things, which all play a role in healthcare as a business, but it's still about just delivering good quality care to people and making sure that people are getting better, and we're doing that in a timely, responsible fashion. I mean, I think that's the underlying principle for just about every department that we look at least that's the idea. Oh, there you are, Jean. So, Jean, actually, I didn't hear your audio right there.
We're back, at least in part.
Okay, just making sure. So,
no, for technical staff.
Yeah, we are first and foremost wanting to make sure we're communicating with patients and that patients are hearing us and that we can understand back what they need. So trying to talk to a patient in an office setting is pretty straightforward, but how they get into the office can be a problem sometimes. And also, once they have a problem after they leave the office or the hospital after their procedure or their admission, we have to figure out how we can make sure that they're still doing okay. Lots of times they have questions, lots of times they can't get supplies they need, a prescription isn't available or something like that. And we don't hear about it for a little while, or it gets lost along the way. I've had more than one patient come up to me and say, Dr. Kennedy, I've been trying to reach your office for 2 weeks now, and they keep saying you're gonna call me back and you never call me back. I don't have a single message. And it probably got lost somewhere in the communication system in our hospital. We've got an EMR that's extremely well used throughout the world. Everyone knows it. And it's a great platform. It has a lot of bells and whistles, but we still find that things break down. So trying to make sure that we hear what our patients are telling us, what kind of problems they're having, and making sure we're giving them the answers that they need in a clear and timely fashion can be a real issue. So,
in terms of your experiences and your colleagues' experiences that you've talked to, you mentioned the EMRs and problems of communication. I think, at least in my understanding, would sometimes come down to the tool is or isn't there, but other times the problem of communication would come down to personal approach or training or things like that. Other times it comes to bad processes that haven't been really thought through or laid out, so there are places where balls can get dropped. Have you received any insight onto which of those seem to cause most of the problems, or maybe you could share some anecdotes about ones that tend to be recurring problem points?
Yeah, that is a perfect way to tee it up there, Jean, because one thing I have found is everyone is saying, we have this great digital health app that's going to make it fabulous for patients. The problem I see in vascular surgery is that most of my patients, not as much these days, but especially when I was doing arterial work, they tend to be older. They tend to not have access to technology, or they have a cell phone, but it's a flip phone and they leave it turned off in the glove box of their car, and that's where it lives. So you can have this great technology, but if your patient can't wrap their heads around exactly how to use the technology, it's not really going to work. The other thing that I've actually had a few different companies mention, and I'm just going to keep it kind of high level there, it has to do with communication barriers that patients have. People who are hard of hearing, people with language barriers, people who have difficulty with speech or typing. When somebody leaves the office or leave the hospital, they don't have the benefit of whatever resources we may give them. And they have to still follow those directions and ask those questions out in their community. And sometimes that can be a challenge to get that information back to the physician or to the nursing pool or whoever's fielding those calls in a way that we can really help them.
It's interesting because I know in our own business, when COVID hit, we often find ourselves in sort of a paralysis of, I've got so many methods of communication, I've got so many ways I could document, that you find yourself, okay, which one should I use? And sometimes you can constrain people down and say, we're going to use this one. But oftentimes there's so many, you want freedom to be able to move around, but the freedom then can create lots of missed places. I know in our own organization, COVID and knowing that we weren't going to be in the same room actually pushed us to better define, this is the pathways that information is going to flow through. And sometimes it almost takes a crisis to be able to say, okay, there are the missed points. These are the places where information can get dropped. Let's make sure we chart those.
Absolutely.
Here's a question for you, though. So you and ArborHive live in a nice bridge point between the world of innovation and the world of the healthcare providers. So 2 questions. One is, if I'm a doctor and I've got concerns about communication inside of a healthcare system, what would be your advice to me? And then the second question alongside of that is, if I'm a startup and I think I'm going to help doctors with communications issues, and I'm thinking a lot of our friends who do digital health and communication systems and stuff like that, what would be your advice to me, given that you see both those worlds on a daily basis?
That's a great question that has a twofold answer.
Okay.
The first one is, oftentimes healthcare systems, at least larger systems, have technology available. Again, the EMR, the health system I work with the most, has a lot of great opportunities for video visits through their platform, different secure texting and sending videos under HIPAA protected channels. That's patient privacy, of course. So our health system has put a lot of effort into providing those tools, because we've been saying for years, we need these things, we need these things. A smaller health system or a private practice doctor may not always have those options, and that can be more challenging. That's, I think, a great place where smaller third-party developers might be able to really listen to what those docs are having to say, and seeing if there's a way outside of those giant EMR denizens of communication these days, to try to find a way to get something to fit in there. I find a lot of the problems that we have are that the EMR companies tend to get a little proprietary about what they have. So they will sometimes collaborate with startups who have great digital health platforms and add that into their arsenal or into their library, but not always. Sometimes they say, we have our own orchard that we're growing, and that's what we're going to stick with. So it is a little bit of a IP barrier at this time, so it can be challenging. I think from our standpoint at our health system, we did surveys, we did a lot of outreach, and the overwhelming cry was that we need a better way to communicate. And one of the things we did is we actually went in and bought a second system in addition to our EMR that allows for secure texting, secure calling, secure video, and that has made a big difference for us. So,
that's very cool.
The second part would be more for the startups, and that is, you need to probably go and talk to a lot of doctors. It just comes down to customer discovery. I've met a lot of great startups with some brilliant people and some amazing technology, but sometimes it's just not what doctors would use, or sometimes it's just not something that a patient could figure out. I've been in that same boat myself, trying to come up with some projects, and then I realized my patients, again, can't use it because they don't have a cell phone or they don't have reliable internet access. And that's not something I really even thought of until I started actually talking to patients and kept coming into these barriers all the time. So knowing which customer segment you're going to target is really critical for these startups. I'm sure that's something that they've all heard before, but especially in healthcare, because it is such a strange and nebulous world to live in sometimes. So, but doctors mostly are pretty happy to complain when there's something they don't like. So it should be pretty easy to find different physicians who are willing to talk about their own personal experiences, which, incidental plug for ArborHive, is what we try to do, is find docs who can connect with small startups and other groups to try to help give them some insight on a friendly basis. But I feel that having those conversations and really understanding the problem that you're trying to tackle in the healthcare world is crucial when you're getting into, especially a digital health startup business.
Yeah, I'm understanding that customer segment so important, and even more so there. You know, what we said we're going to do with From the Frontlines is we've got one more topic to talk generally about, which is going to be the compliance question, which we're going to do
Oh,
the next
yes.
time you're on. Now we're going to start to unpack each of these and start to talk to some docs and represent some more specific things. Non-incidental plug, if you're a healthcare practitioner listening, please go out and make contact with Niki and ArborHive to become part of that discussion, right? It's important for us to have all these general discussions together as innovators and manufacturers and funders and developers, but it's also important to get there with doctors like yourself and really to be able to understand each other's mentality and make suggestions and say, we could do this next, we could do this next. So I hope people do that. I do think this is going to be an interesting one, though, to talk about more, Niki, because communication, we're just inundated with communication methods. There's so many out there in the public domain. Of course, HIPAA brings this next layer of challenge on there. And company policies bring this next layer of challenge there. It's a place where there's room for innovation, but man, a lot of that innovation has to be, like you just said, it's got to understand what that market segment really needs.
It's so much harder than anything else and makes so much less sense than anything else when it comes to things like privacy and security and communications and billing and documentation. It really is its own world when you're talking about healthcare, but there are a lot of us who have been living it for a long time, and we're happy to try to maybe not explain why it is, but at least explain how it is. So.
And that is so important, just understanding what the landscape is. And that's what From the Frontlines is about. That's what ArborHive is about. And we are so glad to have you on MedTech Crossroads. We're looking forward to the next installment in about a month. Dr. Niki Kennedy with ArborHive. Thanks for being with us today.
Thanks so much, Gene. Appreciate the opportunity. We'll see you next time.
Look forward to it. Always love hearing from Niki. She was a frequent participant back when this was COVID Connect. And then as we switched to MedTech Crossroads, we thought, wow, we want to hear doctor perspectives. And I think communication is a huge one. Next up, I want to thank him for his patience and welcome Eric Icard from The Right Place over in Grand Rapids, Michigan. Eric, how you doing? Welcome to MedTech Crossroads.
I'm doing great. It's a beautiful day. So
It's
last
a
thing
beautiful day.
in my yard work. So
Oh, I'm sorry. Well, we'll be talking for a long time then. We'll keep you from that.
that's all right.
Eric, you have been doing amazing things on the west side of the state there. And of course, when we think of West Michigan, we think of very mature businesses, many manufacturing businesses, deep networks, a lot of very mature businesses. And I know you had mentioned to me when we were preparing for this that what we wanted to talk about today was you guys have a lot of like industry councils and things like that you've had, and then here comes COVID, and it hits like it hits all of us, and you guys are going, oh boy, what do we do? You were able, interestingly, to turn to some of these industry councils to get some of that networking happening for a response. So I guess before we go to what was that response and what sorts of things did you see in West Michigan, first of all, tell us just a few words about The Right Place so everybody knows what it is as an organization. And then tell us about these industry councils that you guys have over there.
Absolutely. And thank you again for the opportunity to be part of this presentation, part of this network. So today I did want to talk about collaboration and really how it helped West Michigan during the pandemic. Now, my job at The Right Place, we're a regional economic development corporation serving West Michigan. Our mission is really to drive economic prosperity for our region. And the entity itself was born out of necessity during the recession of the 1980s. And as a result of a study in the early 80s, 14 area business leaders led by Jay Van Andel each kicked in $100,000 to form this new entity that would really kind of help boost the economic vitality of West Michigan. And since 1985, we've been able to assist in the creation of 50,000 new jobs for West Michigan and billions in new capital investment. And these are projects where we actually had a hand in. We do not count it unless we touch it at The Right Place. And The Right Place itself, by its very nature, is a result of collaboration that I believe is very unique to our region in West Michigan. And as for me, I have a master's degree in economic development. I have worked in the industry for 20 years in various states. And I've been serving as the senior business development manager at The Right Place since 2013 for 7 years. And my roles include working with the med device, life sciences, aerospace, and defense industries and companies here in the region. And I also run My Device, which stands for MI Michigan Device, which is our regional medical device consortium. Well, looking at, as we mentioned, the industry councils, that is something that we as an organization have been doing since almost our very beginning. We recognize manufacturing as the most important component of our economy in West Michigan. And in 1989, the Right Place started the Manufacturers Council. And since that time, we've actually started other councils that we've felt have play a specific role in various industries, including the Supply Chain Council. Since we do have such a huge powerhouse of supply chain educational institutions in West Michigan, well, in Michigan in general, it's been a very successful council. We also have the Technology Council, which is a rapidly growing council, and of course, My Device. Both the Technology Council and My Device are also supported by the Grand Rapids Smart Zone. So we do have funding through our membership dues, but through the Smart Zone, we're able to do some unique programming and actually network, or I should say market West Michigan, the assets of West Michigan to companies across the globe.
That's great. Now, so the thing with councils that I often think, okay, well, you've got people there, and that's amazing, and you do the herding of the cats to bring them to the table and say, hey, would you be willing to be part of that? And that's great. One of the things that I think often gets missed is how do you, under normal circumstances, we're going to talk about COVID in a second, but under normal circumstances, how do you leverage the knowledge of those councils? So who participates in them, first of all? But then you got all these people in a think tank. How do you actually get anything out of them? How does that work?
Well, you know, the councils themselves are typically, the companies that participate are invested in the councils, I'll say that. And it's typically the CEO or the COO of the company that's playing a role in active participation in the meetings. These groups typically get together every other month at a host entity. One of the members will host the group. They'll have a plant tour. They'll give a presentation on their enterprise, and then they'll talk about something that's specific to their specific niche. And lately, on the Manufacturers Council side, for example, you know, Industry 4.0 has been a huge issue, a topic of discussion, as well as talent, more so obviously before COVID, but it seems like it's going to be continue to be an issue. And they also talk about best practices. And in the case of the Manufacturers Council, you know, you look at the size of the companies, you have companies that are small as 5 million in revenue to companies that are well over 10 billion in revenue. The My Device Medical Device Consortium, we have companies that, you know, in that 5 million range up to about $1 billion in our membership. So it really provides a forum for, you know, and sometimes in some instances, fierce competitors like a Steelcase and a Herman Miller to come together and try to better their businesses. So I think it's something indicative of West Michigan that we have that type of entity.
So the council as much speaks amongst itself as it does to folks who may be up and coming. It's as much a chance for those groups to talk with each other as well as to offer their resources to newer folks in the community. Exactly.
And you know, if I'm a company with 50 employees and 5 million in revenue, and I'm hearing from a seasoned business leader that has many billions in revenue and thousands of employees, you don't get that kind of access to that type of individual, especially on a peer-to-peer basis, which is, I think, a unique circumstance.
Yeah, that's a wonderful thing. Well, and so these were working for you well before COVID, and then here it comes. You guys, you know, just like everyone else, had to respond to this, and I think you, like me, we found ourselves standing at desks and just kind of chugging and connecting people. And how did the councils work out for that? Because you had something built in there that you could leverage.
Well, the way we approached it, is like we often do in this area. We didn't wait for the federal government to react and to tell us what to do. With the guidance of the right place, we pulled together leaders in the healthcare systems, with manufacturers, and with academia, including Michigan State University's College of Human Medicine and local government. And because of my role working with the medical device companies, I was part of a team that included the Kent County Emergency Operations Command, the area healthcare systems and providers, and that was kind of the core group, I would say. So as they identified needs, mostly regarding PPE, it was, and other things, but it was our job to really see what we could do, and looking at the capabilities of those manufacturers, those council members, to see if we could get them to step up and fill those needs. And we were very successful in doing a lot of that. So, and looking at some of the companies and what they were able to do, just in the medical device consortium, we had them partnering with GM and Ford on the ventilators. We had companies making sanitizers. We had companies making hand sanitizers, gowns, face shields, face masks, goggles, test swabs. And we were even able to leverage one of our members of My Device that's based out of Battle Creek. Battle Creek has a huge runway and a foreign trade zone that's probably underutilized. So rather than fly the shipment into LAX and clear customs there, they flew it into Battle Creek, and they were able to offload a lot of PPE.
Wow.
Otherwise would have been, the government was often holding up. So it's interesting how all these enterprises came together. And
convenient to have. And it sounds like you saw a lot of, as we did everywhere, a lot of gaps that needed to be filled. I remember when the whole thing hit, everybody thought, well, I'm going to try to help. And many of us, the way that we thought we were going to help wasn't the best way to help. For us, it turned into what was the originator of this webinar series, which was a way to connect people and to form a communications channel. You guys were able to leverage these things. Give us some examples of some gaps that you guys found and were able to fill.
There were kind of countless. It seemed like we'd get something new every day. I'd get many calls from the Kent County Emergency Operations Command, everything from even grocery bags to feed families, to fill with food, and specimen coolers. It always seemed like it would be on a Saturday morning when I was hoping to get a little relaxation that we would get that call from Matt at Kent County. But what we ended up doing on the weekdays anyway is we had a med supply team, and we would have a daily meeting. And that included Sam Zablecki with the Michigan Economic Development Corporation, as well as the members of the Michigan Manufacturing Technology Center West. And that entity in itself really gets into the details on the capabilities of area manufacturers. And they have this huge nationwide network. So as we saw needs come in that we weren't sure what we could do, MMTC West was really able to help with that and connecting those suppliers. They were just awesome partners, as was the MEDC. And we saw a lot of sobering needs that we were able to make connections to, and I hate to say it, but from refrigerated trucks to field hospital equipment, et cetera. So
yeah, it was important to have people making those connections, and we're all thankful that you guys were there to do that. I know that you guys got involved in the whole swab thing. We had Jim Metzger from Keystone on. Mm-hmm. Any examples from those kinds of efforts?
Well, Jim has to be my favorite example. When I look at, out of all the things that could have aligned, is that one, and there's another example with the sample coolers. But Jim's was interesting because I'm married to a physician that works at Helen DeVos Children's Hospital. So when I was told that there was this huge shortage of swabs, I was not aware of it, and then confirmed it with Spectrum Health and confirmed it with the Kent County Emergency Operations Command. Both of them were saying, yeah, we have less than 200 left in our inventory. Or to get additional ones. So 21st, and saying basically the lines of, Jim, do you know what a nasal pharyngeal swab is? And responds, isn't that like a stick with some foam at the end of it? I said, I think so. He says, it might be something else. You think you can make it? And his response was, we can, let's do it. Do you have any specs? So after a few calls, we were able to get some samples and some specs in Jim's hands. And, you know, within 6 weeks, Keystone was supplying swabs to West Michigan, well, actually across the state and now New York State. But
oh, wow.
Had I not been to Jim's facility for a My Device tour and meeting and learned about his capabilities and known that, you know, Keystone is a very nimble company and they have both the product development side, product development experience, as well as the manufacturing of disposables, I would not have known that. And Jim really pulled off a miracle. And in true West Michigan nature, he also partnered with other companies that are part of the consortium to make other products. So it's kind of a neat story. I look at another example on of course, a Saturday morning. That's
of course a Saturday morning, yeah.
And, saying we need specimen coolers, and I had no idea what a specimen cooler looks like. I'm an economic developer, so he sends me a picture of one, and I'm like, I have some Omaha State coolers in my garage. You're welcome to have them. But that was about the best I could think of. And then, you know, I recalled what had happened when I saw the picture. We coincidentally, serendipitously, I should say, we hosted the royal family of the Netherlands in 2015. One of the members of that delegation was a new mom who had to ship breast milk back to the Netherlands and needed a cooler. We happened to be at the Van Andel Institute at the time. They were hosting a reception, and I recalled that, and I texted my CEO. I said, reach out to Dave Van Andel. They have these coolers. And by the end of the day, we had the 300 specimen coolers that we needed. So it's kind of amazing how these little things work and all come together.
Yeah, it's really amazing. It's so cool that you guys were able to do what you did, and so neat to see how you didn't have to scramble for the basic underlying communications networks and councils and things. The people were already connected, which I think is a great testament to what you guys are doing out there. Take just a minute, Eric, and tell us about the Right Place. And as hopefully at some point here we get back to kind of normal life, what should people be reaching out to the Right Place for?
Really, we like to think of our as the central point in West Michigan for anything business related, any services, any connections that you would make. I get interesting calls. I had an individual asking me what's the traffic count on 28th Street between these two streets. I've had a person contact me saying, you know, I make medical devices. I need to test them on something. We need cadavers. Where can I get cadavers? So I say anything. I really mean anything. If there is a need and we'll find the answer for you. We have a terrific research capability within our organization and a passion to make sure that businesses are supported.
That's great. Well, we appreciate what you're doing and appreciate what you were able to do during COVID. Hope you're getting a little more rest now and your Saturdays are somewhat more your own. And we are glad to have you here on MedTech Crossroads. And I hope you'll come back and keep us abreast of what's happening in West Michigan. And as folks come up that you think we should be talking to, we'd love to have those recommendations.
I'd love to. Thank you.
Thanks for being with us, Eric. Really appreciate it. I want to take just a minute. We usually open up for Q&A at the end of our show, and because we had a couple of questions we weren't able to get to, I want to just I believe we've still got Dr. Niki Kennedy and Dr. Duane Newton on the line, and I want to just see if we can address two questions that had been sent through that we didn't get a chance to talk about here. Maybe we can start with Dr. Newton, if you're still there, Dr. Newton. This was the question about hand soaps Olaf's question was, there he is. What do you know about antimicrobials that are effective against SARS-CoV-2, that is disinfecting surfaces, hands, face with wipes that have an antimicrobial included, like a liquid, maybe isopropanol or Microban or chitosan? And there was a little bit of chat in the Q&A I thought you had some interesting perspectives on that. I wanted to make sure that we heard you out because I think this is sort of public health, public
Yeah,
health knowledge.
for sure. So thanks to Steve for finding in the approved list from the EPA. I think that was, it's always great to have real information in addition to philosophical information, which I'm going to provide. So Steve gives you the facts and I give you sort of some more big picture perspective. So one of the things to realize is that the antimicrobial soaps that you can buy in whatever store, and those have antibiotics in it that are active against bacteria. They're not, they don't have any antiviral properties. And so there's a lot of debate actually about the public health value of those antimicrobial agents and whether or not it's actually promoting or promoting antimicrobial resistance in populations of organisms in the environment. And so those are targeting bacteria that might be present on your hands, but it's not going to do anything against, those drugs are not going to do anything against viruses. The physical aspects of washing and doing it, singing the ABCs. At Michigan, we sing the fight song when we wash our hands, but that's the amount of time that you're supposed to take, and so that physical nature of that is beneficial. And actually, the detergent that's present in soap can have there's some viruses that their outer envelope will be destroyed because of the detergent, and so that has some antiviral properties. The other chemicals that were listed, that Olaf listed, and that are present in the list, do have specific antiviral properties, and that includes alcohol-based cleaners and wipes and bleach-based cleaners and wipes. The benefits, though, are time dependent, and so it's not just the physical activity of wiping things down, but it's the exposure of the organism to that chemical for a certain amount of time for it to have the intended effects. And so all those things need to be taken into consideration. And we have a lot of different products that we use in our laboratory to ensure that we're cleaning and disinfecting surfaces and decontaminating them so that we protect ourselves and protect the integrity of the specimens from our patients.
It sounds like just simply washing your hands and spending the time doing it. Nothing's quite so good as just beating them up the traditional way.
Well, and that goes true. I think that's how the outbreak is going to go away, is those standard things: social distance, wear your mask, and wash your hands. And as long as we're all doing that, we're going to be protecting ourselves, but more importantly, we're protecting each other.
Well, we appreciate that extra insight there, and it's a good reminder to sometimes there are some good traditional ways to do things, and we can knock those out through that. We appreciate that. I want to bring Dr. Niki Kennedy back for just a moment, because there was a specific question that got asked, which is examples of critical communications that are sometimes missing. At what level was that? Was that like we disagree with government policy, or was that a specific patient advice? You had an interesting answer in the chat I just wanted to bring, because people won't always see the chat afterwards in the recording. But did you want to address that real quick, Dr. Kennedy?
Sure. So I think Olaf's question was, as I interpret it at least, and Olaf, tell me if I'm wrong, are we talking more about our patient and patient-related communications? Or are we talking about, again, something more of like a government policy, payer, that kind of question? In this case, I was referring specifically to information that contains private patient information. There are a lot of different rules along the way. They follow along basically in something called the HIPAA Act. And I'm just trying to look up to see exactly what HIPAA stands for, because it's not something that we always think of. It's the Health Insurance Portability and Accountability Act. So HIPAA, that is something that we all deal with and struggle with in the healthcare world, because it essentially is a privacy rule where we cannot allow patient information to be shared publicly. And that's not a matter of putting on a billboard. That's a matter of putting it in a text message that isn't on a secure texting platform. That's with leaving a voicemail at somebody's house. I mean, some basic stuff that we really rely on as critical communication channels, we can't use as freely. So when we're talking about trying to get in touch with a patient, we can't always tell them their test results in a voicemail because sometimes that breaks some of these HIPAA rules. We can't necessarily text a colleague and say, hey, did Mr. Jones come see you the other day? Because we can't mention Mr. Jones's name or any other identified information in those kinds of communication channels. It can be a real problem when I'm working with some of my ancillary staff. Like I work frequently with a prosthetics officer who helps me with my amputation patients. And we have to figure out some way we're going to communicate about patients, especially if somebody's having a problem, needs to come back in and see me, or if I need to sign some prescriptions because somebody needs to get their shiny new leg. And we can't necessarily just text each other that patient's information openly. So I don't know, I'm hoping that answers Olaf's question, but we do have some significant and unusual barriers that probably don't exist in other worlds, probably in some level of finance and accounting and some legal, but it is a little strange, some of the things that we're not allowed to say if you're not used to thinking of it in those terms.
You have to watch those barriers and make clear what you say, and
Oh, yeah.
we're so used to combining our professional and work, and everything sort of blends, especially now that working from home and stuff like that. I did make Olaf's mic live so he can tell you if you nailed it there and answered his question. And while he's coming live, I just want to let folks know if you have a question that you want to ask about anything we've talked about today or on a previous show, now's your chance. Raise your hand. You can pop it up there and we can chat about it. Otherwise, we're going to give Olaf the moment to say whether we got or if he had any other questions. And then after that, we're going to get going here. So Olaf, are you there?
Yeah, great. Thank you, Dr. Kennedy. That answers it. And I'd worked for a large consumer products company, and there also it was always a question of like, you have email systems, you have other systems for storing records, documentation, and then you have text messaging. And now I understand also the context of what you're saying is like for smaller companies, they might need something customized that includes all the needs that they have. So you don't use Google Messenger to ask about a particular patient record or something just because you need to reach another colleague in a hospital or so.
I mean, it's a huge problem. Absolutely. No, it's you really got down to a very specific problem that is a huge pain point for a lot of, especially smaller physicians or private practice doctors who may not have access to some of the nice technological toys that the rest of us do if we're affiliated with a larger health system. So.
Right. Okay.
Great.
Thank
Olaf,
you.
thank you so much. Dr. Kennedy, thank you. We appreciate having you here.
Thanks.
And once again, to everybody else, thank you for being part of MedTech Crossroads today. What a jam-packed show today. We appreciate Dr. Newton, Dr. Kennedy, and Eric Icard from The Right Place coming on to chat with us. Next week, I've got Tom Evans talking to us about non-contact ECG measurements. And we're also going to be talking about these terms: approval, clearance, granting, authorization, registration, and listing. If you are at all involved in the delivery of MedTech products, product development with FDA, you're going to not want to miss that one, because the pathway that you're going down is going to differ depending on a very few words that have a whole lot of meaning. We're going to unpack that next week. So for me and the whole team, thank you for being part of MedTech Crossroads today. I'm looking forward to seeing you next time. Thanks for being here. Bye-bye.